Bismillah ar-Rahman ar-Rahim, and this is the June 23 paper, and we start a new set of series of all the June 23 exams which were held, and the results have just been declared. Of course, it's a little controversial because some exams might be held. So we start with the paper, the MCQ paper for the A-level exam, and I'm going to discuss all the questions, and maybe this is a new format which I've adopted, is that I'm going to be discussing the correct answer and then discussing the ones which are not the correct answer and why they're not the correct answer. Now, as you all know that since June 22, now the paper is now for 1 hour and 15 minutes, so the difficulty level has increased because previously it was only for 1 hour. But you still have only 40 questions and you have to answer in the MCQ pattern. Now, as you can see in the first question, the basic skill which you must have is the art of reading a question. And that is sometimes lacking in some of the students, and you must try to develop that skill. The electron micrograph shows onion root cells. So the fact is that these are plant cells prepared using a freeze fracture technique. The cells were quickly frozen and then physically broken apart. Freeze fracture breaks apart cells along weak areas such as membranes and the surface of organelles. Surfaces of organelles. So where there are membranes and where there are surfaces of organelles, so it breaks them from there. So there's a label which is structure X. Now that looks like some sort of a little depression. It doesn't look like something which is raised. Now you read the question, it is magnified 20,000 times, that is what is given, so you can understand there's a very high magnification. Now it says, which statement best explains the appearance of the electron micrograph? Now, the cells were broken apart at the endoplasmic reticulum, structure X is a ribosome. The cells were broken apart at the nuclear envelope, structure X is a nuclear envelope, sorry, a nuclear pore. So this structure X is either a ribosome or a nuclear pore. The cells were broken apart at the nuclear envelope, structure X is a ribosome. So nuclear envelope and a ribosome on top of that. Now you know that is incorrect. The ribosomes are on the rough endoplasmic reticulum, they are not on the nuclear envelope. The cells were broken apart at the tonoplast. Tonoplast is the vacuole membrane. And structure X is a plasmodesmata. Frankly speaking, there are no plasmodesmata in the tonoplast. The tonoplasts are in adjacent to two cells, so in the plant cell. Well, yes, we're talking of the onion cell, so there is a plant cell. But why is D wrong? Because tonoplast does not have a plasmodesmata. Plasmodesmata is the cytoplasmic thread between two adjacent cells, right? So this one is definitely wrong. This one is wrong because ribosomes are not present on the nuclear envelope. Now we are left between A and B. Now the question is, how are you going to narrow down your choices? Now cells were broken apart at the endoplasmic reticulum. Yes, endoplasmic reticulum is at a cell membrane. And structure X is a ribosome. But you look at these are depressions. You see they are depressions, they are not something raised. Like this is something raised. This is something raised. So that is why the answer is B. Now I want you to look at some diagrams where I'm going to tell you how I figured this out. I figured this out because, I sort of looked at this. This was something which reminded me. I think it reminds me of a flower. And whenever I look at nuclear pores, it reminds me of a flower. Now just look at this diagram. That's why I tell you, whenever you're studying, you must look at images. Because bio is all about visualization. So you look at these pores. The nuclear pores are very, very interesting. They remind me of little flowers, stuck onto something. So the nuclear pores, this is the diagram which I had in mind when I was looking at this question. And I always remember this very clearly, that the nuclear pores are something like this. And then there's a diagram of this. You can see this here, the details of this here. So that is why the answer was, for the question 1, the answer is B. Now question number 2, which cell structures can form vesicle, cell surface membrane, endoplasmic reticulum, and Golgi body? You know Golgi vesicle, you know endoplasmic reticulum, buds of secretory vesicle, cell surface membrane, phagocytic. When the phagocyte engulfs a bacteria, that's called a phagocytic vesicle. So the answer is A. because all three can form vesicles. Question number 3. Four students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. 1. Synthesis of polypeptides. Number 2. Synthesis of lipids. Number 3. Packaging of hydrolytic enzymes that will remain in the cell. So that means formation of lysosomes. The appearances were listed by letter V, W, X, Y, Z. Which student correctly matched the numbered function with the appearance of the cell structure? Now you know synthesis of polypeptides is on the ribosomes. So what are ribosomes? Ribosomes is W, non-membrane bound spherical structure. So W has to be 1. Then number 2 was synthesis of lipids. That is what? That is smooth endoplasmic reticulum. So smooth endoplasmic reticulum is membranes which surround an enclosed inner cavity. So the answer is B. Then number 3, packaging of hydrolytic enzyme that will remain in the cell. So membrane bound sacs arranged as a flattened stack. So Golgi body is the one which results in the lysosomes being formed. So the answer is A. Question number 4. So which cells contain a tonoplast? Now we know root hair, companion, sieve tube element, and endodermis. Now the only one which does not contain a sieve tube element is the what does not contain a tonoplast is a sieve tube element. So that is why the answer is D. Root hair cells also contain a tonoplast, companion cells also contain, and endodermal cells also have a tonoplast. So all of them except for the sieve tube, which has no tonoplast and there's just a little bit of peripheral cytoplasm. Then question number 5, which organelles found in animal or plant cells are surrounded by double membrane? So question 5, you know the answer is B, chloroplast double membrane, mitochondria double membrane, and the nucleus is also a double phospholipid bilayer. Please understand that. Chloroplast have an outer inner membrane, mitochondria have an outer inner membrane, the nucleus has a double phospholipid bilayer. So they are 1, 2, 3, 4 phospholipid molecules. Question number 6. Some scientists think that mitochondria evolved from bacteria that entered the cytoplasm of a different cell and were able to survive there. So this is a cell and the bacteria entered it. So which structural features of mitochondria support this hypothesis? Why? Because you see mitochondria contain circular DNA and 70S ribosome. But the folded internal membrane, so bacteria does not have any folded internal membrane. So please remember that. So that is why the answer to question 6 is D. So the folded internal membrane did not come from the bacteria, but bacteria does not have a folded internal membrane. The outer membrane of the mitochondria is this, and the inner membrane is this, and it contains DNA and 70S ribosomes. So DNA and 70S ribosomes also are present in bacteria. So that is why the answer is D. Then it says, what is present in all viruses, all prokaryotes, and all eukaryotes? Now, it's a very interesting question because you see, you have to relate virus, prokaryotes, bacteria, and all eukaryotes. That means animal and plant cell and fungal cells. So, ribose can be present in viruses. Why? Because viruses can be RNA or DNA. Prokaryotes can also have, bacteria can have mRNA, so ribose can be present, eukaryotes. But what is present in all? You have to understand is what is present in all. It didn't say some viruses, it says in all viruses. So we said viruses are of two types. Some have DNA, some have RNA. But it said what is present in all viruses, all prokaryotes, and all eukaryotes. Now this is a little interesting question because ribose and deoxyribose, DNA and RNA viruses can either have DNA or RNA. They cannot have both. So you have to understand is this thing. So ribose is incorrect. Deoxyribose is incorrect. Now the only correct thing which is left is, you see, you have to choose between cytosine and thymine. So the answer is C. But why is the answer C? Because cytosine would be present in DNA and RNA. Thymine is only present in ATGC, is only present in DNA. In RNA, it's AUGC. So it's a little trick question. Difficult question, I would say. I would say some students would get this right, large majority would get it wrong because they did not read all viruses. So viruses, whether they have DNA or RNA, they will have cytosine because you see, you have to remember is DNA contains what? DNA contains ATGC, while RNA contains AUGC. So basically G and C is common to both, and A is common to adenine, guanine, and cytosine is common to both DNA and RNA, and it said all viruses. So the answer in another paper, they could give you also adenine or guanine or adenine. Thymine is only present in DNA. So this cannot be there. Question number 8. The table shows some steps that can be made in carrying out the Benedict's test. Which combination of steps is required to carry out a semi-quantitative test on a reducing sugar solution? So why would you boil it with hydrochloric acid? That's not a reducing sugar, that's a non-reducing sugar. So this should not be done. So that is why the answer is C. Why would we do a test of hydrochloric acid and neutralize with alkali? You only do that with sucrose. Sucrose is a non-reducing sugar. Here it said you just have to do a quantitative test on the reducing sugar. So this one was wrong. This was only done when we do a non-reducing sugar. But if you don't know it, well, of course, if you haven't revised your syllabus properly, then you're probably going to mess it up. Question number 9. The diagram shows 3 examples of different bonds. Bond 1, bond 2. Bond 2 is a peptide. Bond 3 is a disulfide. Bond 1 is a hydrogen. Which bonds hold the secondary structure of proteins together? So that's hydrogen bonds. So the answer to 9 is C. Only 1 is the one because the hydrogen bonds will hold it. You see, the peptide bonds are in the primary structure. The hydrogen bonds are in the secondary structure. Then coming to question number 10. Insulin is a globular protein involved in cell signaling. It is transported in the blood plasma from the cell that synthesizes it to target cells. The molecule of insulin contains 6 sulfur-containing amino acids and has 2 polypeptide chains. Which statements about insulin are correct? An insulin molecule has a quaternary structure. Insulin polypeptides are held together by 6 disulfide bonds. This is here where the trick question comes. You see, if this is a amino acid with sulfur and this is an amino acid with sulfur, and you have 6 of these, so there can only be 3 disulfide bonds. So this was a good question. So question number 10, the answer is C. Why is the answer C is 1 and 3 only? Why? Because an insulin molecule has a quaternary structure because it has 2 polypeptide chains. Quaternary means 2 or more. And amino acids with hydrophobic R groups would be found in the center of the insulin molecule. Why? Because it's a globular protein. Globular proteins have the hydrophobic R groups in the center and the hydrophilic R groups on the outside. So this is what you have to understand. It cannot be having 6 sulfur containing and has 6 disulfide bonds. So this was the thing which you have to understand if you are clearly going to read it and understand the question. Then question number 11. Which statement correctly explains why blood plasma can be maintained at a stable temperature? Please remember, your blood plasma is the fluid inside your blood vessels. So why is it maintained at 37 degrees Celsius all the time? It has a low specific heat capacity, high specific heat capacity, low latent heat of vaporization, high latent heat of vaporization. But they are asking you maintained at a stable temperature. So you have to understand is high specific heat capacity. It doesn't sort of fluctuate. The temperature does not fluctuate. If you put your watch under the sun and put it out in the sun, well, it's going to get very hot because iron metal gets very hot. But if you put a Nestle bottle of water outside, it's not going to get that hot. So high specific heat capacity. So the answer to 11 is B. Now question number 12. Which region on the graph shows the activation energy of an enzyme catalyzed reaction? Now you got to understand is that they had given you these. So this is without the enzyme is this one which is got a higher activation energy. Right? And then this one is the one with an enzyme. That's what is labeled here, with enzyme. So this has to be the activation energy. So which region on the graph shows the activation energy of an enzyme catalyzed reaction? So the answer is B. The graph shows the effect of an increasing substrate concentration on the rate of an enzyme catalyzed reaction. And you can see rate of reaction is on the y-axis and substrate concentration is on the x-axis. And this is the maximum rate of reaction which they have given you here. And then you have two graphs, P and Q. Now you can see that P is the normal one and Q there is, of course, is meeting the Vmax, but is reaching it at a higher substrate concentration. So line P represents the result when the enzyme is used at its optimum pH and optimum temperature and without an inhibitor. Line Q represents the result when the reaction conditions are changed. This is usually for a competitive inhibitor. Which description of changes to the reaction conditions could result in line Q if all other conditions were kept the same? Add an inhibitor that attaches to a site other than the active site, that is non-competitive inhibitor. Add an inhibitor that has a similar shape to the substrate, yes, that makes sense. Add an inhibitor that blocks the active site of the enzyme. Carry out the reaction at a higher temperature. So, which one makes sense? Now, what you have to understand is that 2 and 3 only make sense. Why? D is the answer. But why 2 and 3? Add an inhibitor that has a similar shape to the substrate. Yes, this is a competitive inhibitor. Add an inhibitor that blocks the active site of the enzyme for a short while, but when the substrate, you see rate of reaction, this is now the rate of reaction here. So as the substrate increases, then the rate of reaction will reach the Vmax. So if we had 100 active sites and 10 are blocked, but as we increase the substrate, then more chances of the substrate fitting into the ones which are available there. So that is why the answer was D. Question number 14. Which statement about cell signaling is correct? One type of receptor molecule will recognize all ligands in the body. Oh, well, you've got to understand is ligand is a cell signaling molecule. Now, it would have very specific receptors which would have complementary shape to them. Like if insulin has this shape of a triangle, of a rectangle or a triangle, well, then it's going to fit here. So it has to be a complementary. One type of receptor molecule will recognize all ligands in the body. No, that's wrong. The binding of the ligand may cause a change to the shape of the receptor. Yes, that is correct. So B is correct. But then please understand it. The receptors for ligands are always found on the inside of the cells. No, because you see, ligands are water soluble, so they have to attach to a receptor on the surface of the cell surface membrane. Some of them are not. Some of them can attach to the nuclear membrane or to the receptors inside the cell. The same ligand is made by all of the cells of the body. No, insulin is a different molecule, or other cell signaling molecules all have different shapes and they will have complementary shape of the receptors. Now I just want to revise ligands with you. Ligands are primary messengers and they will attach to a receptor on the cell surface membrane, and you see this is the cytoplasm or the cytosol, and that results in one receptor by ligand binding number 2 signal transduction by a second messenger, and then cellular responses and changes in gene expression. While there's another one here, extracellular fluid, plasma membrane, signaling molecule, cytoplasm, intracellular receptor. So there are receptors inside the cytoplasm, and they will result in something with attaching to the DNA and resulting in transcription or translation or some sort of a response. So just a quick revision on ligands and the fact that there are two types. They won't attach. They don't have all receptors on the cell membrane. There's some ligands which will attach because they will pass through the hydrophobic part of the cell membrane, and they will attach to maybe a receptor inside or maybe even to something on the nuclear membrane. Question number 15. 4 students A, B, C, and D observed plant epidermal cells that had been placed in a concentrated sucrose solution. So we've got the plant epidermal cells, and outside you have a concentrated sucrose solution. Concentrated means it has a lower water potential. The inside has a higher water potential. They were asked to identify the partially permeable layer and to explain the appearance of the cell in terms of water potential and movement of water. Well, of course, it has to be the cell membrane. It can't be the cell wall. So this is all rubbish. You don't have to even read those C and D. Now, the cell contents have a lower water potential. No. Why will the cell contents? Because we said concentrated sucrose solution. So concentrated sucrose solution means lower water potential outside and higher water potential than the sucrose solution. So the cell contents will have, and then the water is going to move out. So that is why question number 15, the answer is B. Cell contents have a higher water potential than the sucrose solution. More water moved out of the cell than moved in. You see, you have to talk of the net movement. So water will always move in and out. Some will move in, some will move out. But then what is more? If more water is moving out and less is moving in, then you know it's going to cause plasmolysis. Question 16. The table compares the surface area to volume ratios of 5 agar blocks that differ in dimensions but which all have the same volume. You can say they all have the same volume. This column is the same. The agar blocks can be used to measure the efficiency of diffusion, where efficiency is measured as the time taken for a dye to reach all parts of the block. You know that the rate of diffusion is dependent on the surface area, the concentration gradient. So what we have to understand is where has the surface area the maximum, where the surface area has been 4:1. Now, how has that been achieved? That has been achieved by 32.5. So it's only 0.5 thick, but it's big, 32. The size is big. You see, look at this, 8. That's nearly a cube. But then look at 32.5. So 8 would be a cube. But what would be, so you have to see what would be this one, 4. 32 would be something very, very big, but it would be very, very thin. Because it's only 0.5. This was 8 height. This is 0.5 height. So you've got to understand how they're answering the question, how they're asking the question, where efficiency is measured as the time taken for a dye to reach all parts of the block. So which prediction can be made about the way in which size and dimensions of these blocks affect the efficiency of diffusion? A. The efficiency of diffusion will decrease as the width of the block increases. The efficiency of diffusion will increase as the height of a block increases. The efficiency of a diffusion will increase as a block of fixed volume is flattened. Block of a fixed volume is flattened. The efficiency of diffusion will decrease as a block of fixed volume is elongated. So, the question is which prediction can be made about the way in which size and dimension of these blocks affect the efficiency of diffusion? So the answer is C. The efficiency of diffusion will increase as a block of fixed volume is flattened. You see, this one has become flattened. Efficiency of diffusion is going to be more because it has more surface area to volume ratio. Question number 17. The cell cycle includes mitosis. What are features of this type of nuclear division? Form cells of equal sizes to the parent cell? No, we're only talking of the nuclear division. Prophase, metaphase, anaphase, and telophase. Forms genetically identical cells. Semi-conservative replication of DNA. That occurs in interphase. So this one is wrong, this one is wrong. So the answer is D. Because we're only talking of mitosis. Mitosis is only prophase, metaphase, anaphase, and telophase. That is the whole cell cycle interphase, if we include that as well, which is G1 and S and G2. DNA replication takes an S phase of interphase. Question number 18. A student observed the cells in the growing region of an onion root and obtained the data shown. Interphase 886, 73 prophase, 16 metaphase, 14 anaphase, and 11 telophase. Now it says which percentage of cells contain chromosomes that appear as two chromatids. Now this whole thing is about 1000. Total is 1000. Now which percentage of cells contain chromosomes that are appear? So it was a little difficult because it says which cells contain that appear as two chromatids. You see, if you talked of interphase, in interphase you can't see anything. They might have doubled in S phase of interphase, but then you can't see them. It says appear. That means you can see them under the microscope. You cannot see them under the microscope. You can only see them under the microscope in what? In prophase and metaphase. So 73 plus 16 is what? 89. So it's 8.9%. So the question which was challenging was, I mean, you would have thought of interphase, they've also doubled, but then it says which that appear as two chromatids, that you can actually see them under the microscope. Well, you can't see the interphase under the microscope. They're thin, thread-like, and then they shorten and condense in prophase. So that is the catch. That was a catch, because you could have made a mistake. I know anaphase and telophase, they have now become single. But then if you added the 886 and the 73 and the 16, you would get a different percentage. So remember that. That was a catch in this. The question was appear. Angrezi hai sari. It's all English. It's good English. It's not biology actually. Question number 19. Which statement about messenger RNA is correct? In eukaryotic cells, mRNA is made by removing exons from the primary RNA. No, we remove the introns actually. So this is wrong. Then mRNA is a single-stranded polynucleotide containing a different purine base. No, it has uracil instead of thymine. Now adenine and guanine are purines. The others are pyrimidines. So if you didn't know that, well, you get that wrong as well. mRNA molecules contain ribose sugar joined to phosphate groups by phosphodiester bonds. Yes, so 19C is correct. But why is D wrong? The monomers of mRNA contain a phosphate group and deoxyribose. This is rubbish. RNA does not contain deoxyribose. DNA contains DNA deoxyribose, mRNA, RNA, ribose. So this was wrong. Which structure are involved in transcription only? DNA template strand, anticodons, RNA polymerase. It was a very basic question, nothing to be very ambiguous about. There are no anticodons. So DNA template strand and RNA polymerase is needed. So the answer is B. Next, question number 21. One gene provides a code for the production of which type of molecule? One gene provides the production of proteins, not amino acids, not DNA, not nucleotides. So the answer is D, which is a polypeptide.